Thank you so much. It's really a pleasure for me to be here in the context of the Grand Rounds. This is a kind of a storied series, and I myself have logged in to enjoy several of the presentations. This sounds like it might be a little loud. Is that okay? Okay, and also welcome to everybody who's here online. I thought I would tell you just kind of the backstory, actually, behind this concept of bioorthogonal chemistry. And so the first part of this presentation will be somewhat historic, kind of give you kind of a view as to how the concept of the chemistry came about and what were the motivations. And then I'll finish with one of the more recent applications that is clinically relevant now, and it's actually in the context of antibody drug conjugates, which thank you for mentioning that. So I'm going to come to that at the end of the talk. So let me just start with kind of a high-level view of bioorthogonal chemistry and the backstory. And it started like so many projects start in my lab, which is the identification of an unmet need in biology that we thought we could solve with some technologies from chemistry. And it had to do with inventing chemistries that could be performed in living systems. So just let me start with the contrast of more traditional chemistries. So when I was a graduate student and a postdoc, and even still today in my lab, most chemists think of doing chemical reactions in vessels like this one. This is what we call the round bottom flask. It would be in the fume hood of a chemist here at Stanford, for example. And this vessel is designed so that the chemist has complete control over all of the parameters of the chemical reaction. And by that, I mean the chemist chooses the reagents and the solvent that they're dissolved within and the concentration at which they're dissolved, the atmosphere above the reaction. And sometimes chemists have to purge the atmosphere with either argon gas or nitrogen gas to get rid of oxygen and moisture, which might damage the reaction. The chemist controls the temperature of this reaction. There's a vessel here that can heat up the reaction or cool it down. And by having this much control over all of the components of the reaction, the chemist has 1,000 different transformations they can avail themselves of to react reagents and form bonds and make new forms of matter. But as I became more interested in the life sciences during my training years, it became clear that there was a lot you could do if you could do chemical reactions in much less controllable settings. For example, the setting of a live cell. And why might you want to do chemistry in a live cell? Well, one of the applications would be to attach imaging probes to molecules of interest so that you could follow them around inside the cell within their natural habitat. And in the 1990s, this was a really interesting application of chemistry in living systems, the ability to do chemistry on biological molecules, not just within the cell that they might reside, but even within an organism in the laboratory, like a mouse or a zebrafish. There were some ideas that we had for doing chemistry in these settings to attach probes to molecules of interest. And if you could do that, then why not dream big and think about all of the applications you might be able to pursue if you could do chemistry inside the body of a human, maybe to deliver drugs to a certain place at a certain time, or also for imaging, but now maybe at the clinical level for PET imaging or MRI contrast imaging. And we had this inkling that there might be some really interesting new dimensions of biological research and maybe even diagnostic imaging, if only you could do chemistry in these living systems. So that's what led us to develop a new brand of chemistry, which we gave the title bioorthogonal. And people ask me, what does that mean? And so here's an actual definition. Bioorthogonal chemistry we defined as being chemistry between two reactants that neither interact nor interfere with the biological system in which they reside. And another way to think about that is that in the universe of possible chemical reactions, there are different sectors that a person could explore. For example, in this universe, there is a galaxy that we define as biological reactivity space. And that is the reactivity of all of the chemical groups you would find in biology, right, in our body, for example. And it's a pretty large space, but it's not infinite. And elsewhere in this universe, we thought there would be a different galaxy that we would call the bioorthogonal reactivity space, the chemicals that are orthogonal to biology, that react with each other, but don't cross-react with biology. And that's the space that we have been exploring in my lab. Now, we had a very pragmatic motivation for trying to develop these chemistries for biological settings. And it came out of a different passion of mine over the years, which is the study of glycans, and people call this glycobiology. And I got interested in this during my years as a graduate student back in the late 1980s, early 1990s, because I had joined the lab of a carbohydrate chemist. So I was working on the synthesis of carbohydrates as a graduate student. And along the way, I started reading more about their roles in human biology. And one of the roles that was quite clear, even back then, in the Stone Ages of science, was basically determinants of cell-cell interactions. And glycans are perfectly poised to mediate cell-cell interactions because they are largely found on the surface of cells. So in fact, any cell in your body is coated with complex carbohydrates or with glycans. And it turns out that those glycans form a pattern that's very cell type specific. And even back in the 1980s, and in fact, a few decades before that, people who study cancer tissue and look for molecular changes were well aware that the patterns of those cell surface glycans are altered in tumors. And one of the phenotypes that was quite prominent was the identification of kind of higher densities and higher expression levels of glycans that terminate with a sugar called sialic acid. So it was known and accepted back in the 1980s even that cancer cells had like this overgrown forest of sialylated glycans compared to healthy cells, which have more of a well-manicured garden, I like to think of it, of these sialoglycans. And in recent years, we've actually been studying the reasons why this occurs and the significance of this phenotype. And even we've been targeting this phenotype with new therapeutics. And that is a subject for another day, okay? But at the time that we were thinking about the chemistry and living systems, we thought about this phenotype as something that could be a target for molecular imaging, right? Like any molecular difference between a diseased tissue and a healthy tissue, if you can figure out a way to translate that molecular change into a signal that can be detected or a pattern that can be visualized with imaging modalities, that could be really useful to the practitioner who's trying to monitor, diagnose, detect, and treat these kinds of diseases. So when 1993 or 4 rolled around, I started thinking about my own future career. I was interested in pursuing an academic career and having my own lab. And I was thinking that it would be really great as a chemist if I could figure out a way to image this sugar and detect these kinds of changes non-invasively. And then I went to a conference, and this is one of those stories, like so many of us have, of serendipity, where we happen to be in the right place at the right time and meet the right person that catalyzes an idea, and off we go with it. So what happened was my postdoc advisor had been invited to give a talk at a sort of boutique conference in Southampton, England. And at the last minute, he decided he really didn't want to go. Didn't look like it was much of an interesting conference, and he had regretted accepting this invitation. And so he asked me, would I like to go in his place and give his talk and have a free trip to England? And I thought, yeah, that's a no-brainer. So there I was in this, in fact, not very interesting conference where most of the talks, were not memorable to me, but one of the talks was highly memorable and highly influential, and it was from Werner Reuter, who was a German biochemist. And Werner's lab had been studying the biosynthesis of sialic acid for reasons having nothing to do with cancer. But he talked at this meeting about their observation that that biosynthetic pathway could be infiltrated with chemically altered substrates. So normally within our cell, we have enzymes that convert this precursor sugar called N-acetylminosamine into the sialic acid, which is then utilized by other enzymes so that it can be attached to glycans that are being built in the secretory pathway And the sialic acid ends up on glycans that are on the cell surface, either in glycoproteins or glycolipids. And what they had noticed is that the functional group at this position, which is normally an acetyl group, and for those of you who forgot everything you learned in organic chemistry, acetyl group is what you get when this R is a methyl group, a CH3 group, okay? And that would be the normal substrate for this pathway. But what his group discovered is that this R group could be altered and extended in length conservatively, not too big, but small altered substituents here would be still recognized by these enzymes. And you could actually feed cells a chemically modified monosamine derivative and find that chemically altered sialic acid on the surface of the cells a few hours later. So cells kind of are what they eat when it comes to the sialic acid pathway. And when I heard this talk, it occurred to me that maybe you could capitalize on this phenomenon to deliver a reactive chemical group into sialic acids that you could then exploit in a second step to conjugate an imaging probe chemically. So maybe there was a two-step process of metabolic labeling and chemical labeling that could get you to imaging probes on sialic acids. So as it happens, about 2 months after that conference, I started applying for academic jobs. And when you apply for academic jobs, you have to write up some research proposals and sort of give your search committee a view as to what you might work on in your lab, should they hire you. And so this is a cartoon that came right out of my job applications back in the summer of 1994. This at the time was considered a pretty sophisticated graphic. Today, that wouldn't really be the case. But you can see the idea here was, okay, we're going to image cell surface glycans, and in particular, sialoglycans. And we're going to do that by feeding cells a chemically modified precursor where there's a functional group X. That's right now agnostic. I actually didn't know what X would be at the time, but I thought we'd figure it out. And we'd put a chemical group, feed that to cells, let the cells integrate it into the cell surface glycans, and then feed the cells or expose the cell to an imaging probe with its own chemical group Y, where X and Y were designed to be perfectly matched with each other so that they would form this bond, and now there's a probe on the sugar and you could see the sugar. Okay, that was the idea. But you can see that if one aspires to do this on live cells, in live model organisms, in live human patients, the chemistry between X and Y has to be pretty special. It has to be bioorthogonal, right? X and Y cannot react with any other chemicals that are in the biological system. Otherwise, that's the end of the experiment. They have to react only with each other, and they can't do this in a way that would be toxic to the organism or anything like that. And at the time, I remember spending long weekends and nights plowing through giant volumes, compendia of organic chemical reactions, looking for something that might sort of fulfill all these criteria. And there really wasn't a chemistry at that time that a person could have used in a biological setting. So nonetheless, I did get a job at Berkeley, and I went there and started recruiting graduate students. And our first task was to invent new chemistries that you could imagine using in an experiment like this. And the first chemistry that we developed was modeled after kind of an ancient reaction from the chemical literature called the classic Staudinger reduction. So named because it was first reported by Hermann Staudinger, a very famous German chemist from the previous century. And in fact, Dr. Staudinger was a Nobel laureate in chemistry. He won the Nobel Prize in the 1950s. But way back in 1919, okay, that's more than 100 years ago now, Hermann Staudinger published on the reactivity of these two chemical groups. This one is called a phosphine. That's because it has a phosphorus atom in a certain geometry. And this one is called an azide. An azide is what you get when you put 3 nitrogens together, okay? And the phosphine and the azide will react with each other to form this covalently bonded product, or let's say right now it's an intermediate actually, but it's the product of this first step. It's called an azaylide. And it's a chemistry I was well familiar with because I had used this when I was in graduate school as a tool for chemical synthesis. It's a very mild reaction between two chemicals that are not found in nature. So the phosphine and the azide came from the minds of chemists. They don't exist in the natural world as far as we know. And they react with each other under very mild conditions, physiological pH, physiological temperature. And you can do this chemistry in water, and it won't interfere with anything in a biological system. So it's almost bioorthogonal. And the reason I say almost is because this azaylid is actually not stable in water. So when there's water around, the phosphorus-nitrogen bond gets hydrolyzed spontaneously to give two products, the amine and the phosphine oxide. And the reason it's called the classic Staudinger reduction is because the azide is getting reduced to an amine and the phosphine is getting oxidized to a phosphine oxide, okay? But it's so close. If only the azaylid were stable in water, we could have used this chemistry to take A and B or X and Y and react them with each other and form a bond. And so we started by thinking about ways we could adapt this transformation to form a stable bond. And one of my early graduate students, Eliana Saxon, figured out how to do it. She simply introduced a methyl ester ortho to the phosphorus atom. So what happens is that the phosphine and the azide react. You get that same intermediate, the azaylid, but now this nitrogen atom is situated where it can react intramolecularly with the ester, cleaving the ester and forming an amide bond. And then methanol is ejected as a leaving group. And now, eventually water will still cleave the phosphorus-nitrogen bond, but this cyclization is so much faster than the hydrolysis that by the time water comes along and does its job, the amide bond will hold the red part and the blue part together, even though the phosphorus-nitrogen bond has been cleaved. So we call this the Staudinger ligation, because it actually ligates two things together and the ligation product is stable in water. And this was the first bioorthogonal reaction that we had at our disposal to try to do some in vivo chemistry for imaging purposes. So this is a big, bulky chemical. This is a very small chemical. So we decided that we would put the azide into sugars because the biosynthetic enzymes would be less offended by this small chemical group. And we did that by simply, in the context of sialic acid, we just took a page out of Werner Reuter's playbook and swapped out the acetyl group at this position for an azidoacetyl group. So it's very similar to the natural substrate, just with the addition of this 3 nitrogens. And when we fed this to cells, metabolism happens inside the cell, and then on the cell surface, one would find the azidosialic acid, replacing the natural sialic acids to some extent. And basically, this was a generalizable approach to putting a chemical group into cell surface sugars, not just for sialic acid, but for other sugars like fucose and N-acetyl galactosamine, and basically any sugar that you can find in human cells, and people have also done this in bacterial cells, in plant cells even, and in whole plants. All you have to do is find a place where you can put an azide onto a sugar that enzymes won't be offended by, and let enzymes basically metabolize that sugar and integrate it into these complex carbohydrates. So once the azide is in the sugar, one can then do a reaction, a Staudinger ligation with a phosphine probe. And you could do that on cells. You could even do it inside laboratory mice. So, this was a paper we published in 2004 showing that we could inject the azido precursor of sialic acid into mice and just load up their cells, with the azido version of sialic acid, which was surprisingly quite harmless to these animals. And then later inject into the same animals a phosphine probe that's outfitted for the Staudinger ligation. And the chemistry happens in the body of the animal, and you can detect the product in various ways. And we made a whole bunch of different probes, some with fluorescent dyes on them, some with MRI contrast reagents on them, and so on and so forth. So these were exciting times because, we proved to ourselves and to others that a live animal could be a vessel for a chemical reaction that is so finely tuned that it doesn't interfere with anything else in the animal, and you can actually form a product in there. But we also learned in these early days, in the early 2000s, that the kinetics, the rate of these bioorthogonal reactions is actually a really important parameter to control. And although the Staudinger ligation performed very well in these biological settings, it was a very clean, reliable reaction, it was on the slow side. And when you put your reagents into the round bottom flask in your fume hood, you can be pretty confident that they'll stay in that flask, right? As long as you leave them in there. But when you put your reagents into an animal, which is not anywhere close to an equilibrium setting, the reagents don't just stay in there until the reaction finishes. The animal is metabolizing those reagents and excreting them. And if the reagents are metabolized and excreted faster than the chemistry can take place, you're not going to get much product in that reaction flask. So the clock is ticking on in vivo chemistry, and we discovered that the chemistry really has to be much faster than the Staudinger ligation offered us, probably about 2 orders of magnitude faster. And so we kind of set our focus around this time on trying to increase the speed of a bioorthogonal reaction, and we kind of hit a wall with the Staudinger ligation. However, the good news is that azides, we had chosen the azide as one of our components. We really liked it because it's so small. And azides also do other types of chemistry besides this phosphine chemistry. And you can go back to the German literature of the previous century and find a different mode of reactivity of the azide. And there was work from Professor Rolf Huisken's lab around 1950s, 1960s, showing that azides also do cycloadditions with alkynes, that's a triple bond, to form a product called a triazole. Professor Huisken, he passed away just about 10 years ago, and he was in his mid-90s at the time. I had the great pleasure of meeting him a few years before that, and he was still coming to work at University of Munich every day in his early 90s. And in the previous century, he did a lot of work characterizing the mechanism of this reaction. And one thing we knew from his publications was that, we knew the Staudinger ligation was kind of too slow for us. This chemistry is even slower, okay? So when people do this type of chemistry in the fume hood in their laboratory, they generally have to heat it up. And that's what this symbol means. This is heat. And the kind of heat we're talking about is maybe 100, 110 degrees C, right? And so that's boiling more than the boiling point of water. You can do that in the chemistry fume hood. You can't do that to your cells or your mouse or your human patient, right? So we knew that on its face, this Huisken chemistry was too slow. Now, in 2002, 2 other labs took an interest in azides for chemical synthesis and also recognized the potential of this type of reaction for building complex molecules, and the fact that it would be better if you could do this chemistry at room temp, didn't have to heat it up so high. And the 2 labs that explored this chemistry were the labs of Morten Meldal and Barry Sharpless. And Morten is at University of Copenhagen, and Barry is at Scripps. And totally independently, they both stumbled into a way to accelerate the rate of the azide alkyne cycloaddition, and it's through the use of a copper catalyst. And so if the alkyne is a terminal alkyne, which means that it has some substituent on one side, but just a hydrogen atom on the other, and you add copper, the rate of this reaction goes at room temp from something like 1,000 years, which is without copper, which is essentially never, to 30 minutes with the copper catalyst. And this chemistry is very well tolerated by biological functional groups. So biological functional groups don't interfere with this chemistry. You can do this in water, right? Starting to look like it could be a fast bioorthogonal reaction, but not so fast because that copper catalyst, oh, by the way, they shared the Nobel Prize with me, just in case you didn't know. But this copper catalyzed rendition of the cycloaddition, which Barry Sharpless's wife branded click chemistry. I learned that when we were in Stockholm. They call this click chemistry because it's like you take 2 things together and just click, right? Without any fuss. But that catalyst is something you couldn't use with live cells, live organisms. It's not really suited for in vivo applications. So we knew that as exciting as this chemistry was for, applications in the chemistry lab, it really wasn't going to fly in a biological setting. Now, I teach organic chemistry to sophomores. I teach introductory organic chemistry. Most of my students are pre-meds. Did any of you take my class at Berkeley? Because in an audience like this, I often find somebody who did. I had you for OChem and still got into med school nonetheless. Great. Phew. And when I teach that class, like we all do, there's always a lecture that I give on the concept of ring strain. It's an important concept in organic chemistry. So this is the lecture where we teach our students that bond bond angles, there's certain preferred bond angles for carbon atoms, depending on what we call their hybridization. And if a molecule allows a carbon atom to sit in its preferred bond angle geometry, then you have a strain-free system. And in the context of rings, when you have a 6-membered ring that's all carbons like this, that's comfortable for carbon, and this is essentially strain-free. So these numbers here are the amount of ring strain, and 1.3 kilocalories per mole is very low. But if you start to make the rings smaller and smaller, the bond angles get distorted from where they would prefer to be, to the point where if you put those carbon atoms in a 3-membered ring, you now have 30 kilocalories per mole of strain. That's a lot. And if you try to put double bonds into rings, the strain is even more extreme. And what happens is these very strained ring systems are very reactive, and you can get these bonds to react in ways that they wouldn't if there was no strain. Because if the molecule can release that strain, that's a driving force, and the molecule becomes more comfortable. So I was writing this lecture, because I teach this to students, and as I was writing this lecture back at Berkeley, it occurred to me, maybe you could accelerate the kinetics of that cycloaddition using the principle of ring strain instead of a copper catalyst, okay? And I talked to my students after that class that day, and I said, hey, go to the library and see if has anybody ever done one of these cycloadditions with strained alkynes. This is when you went to the library to look stuff like this up, because there was no internet, or we didn't use the internet much at the time. This is very early days. And so one of my students did that, and he plowed through some old German literature, there's a theme here, and found this paper from 1961, published in a German journal with no English translation. So we had to fight our way through this. And it was from the lab of Georg Wittig, another Nobel laureate in chemistry from the previous century. And in this paper, Wittig had made the observation that azides will react with cyclooctine. So this alkyne is now in an 8-membered ring. And that cyclooctine has about 19 kilocalories per mole of strain. That's because triple bonds want to be linear. And the minute you put them in a ring, they're not linear. They're bent, and they don't like that, okay? And if you made this ring any smaller, it wouldn't be stable at room temperature, okay? So this is the smallest ring you could force an alkyne into where it's stable at room temperature. But it will react vigorously with an azide like phenylazide to form this product. And we knew this because we don't speak German, but you don't need to speak German to understand this sentence, because blah, 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 phenylazide, blah, blah, cyclooctine, blah, 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 explosion, okay? That kind of told the story right there. And we were like, oh, that's very promising. They combined these things and got an explosion. They didn't have to heat it up. There's no copper, So we started digging into this chemistry a little bit more, and we figured out how to make cyclooctines and put ornaments and appendages all over them. To make a long story short, over about 4 years, we discovered that we could make all kinds of cyclooctines and that they would react with azides, and we could tune the kinetics to our desire by structural modifications to the core cyclooctine. So I won't go through all the details on this slide, but I'll pull out a few examples of molecules we made and used, and now many of these are commercially available. Up in this upper left-hand corner is what I consider a vanilla, not very decorated cyclooctine. And it will react with azides, and this is the second order rate constant. And all you need to know is that this rate was comparable to the Staudinger ligation chemistry. Okay, so that was like the floor. And we knew that we needed to increase the rate by about 2 orders of magnitude to be able to play with in vivo imaging applications. And we got pretty close to that by simply putting 2 fluorine atoms next to that alkyne. Okay, we call this difluorocyclooctine, and DIFO for short. And you can see that its second order rate constant is in the order of 76 times 10 to the minus 3 per molar per second versus 1. Okay, so that was right there a breakthrough. DIFO became the first reagent that we really went full force in vivo imaging with. But a few years later, we bumped up the kinetics another order of magnitude or so by fusing 2 benzene rings on either side of the cyclooctine. I call this the West Coast and the East Coast. And those benzene rings increase the ring strain even more. Some people think of this as like the elephant molecule because the ears and the trunk and stuff. Okay. My postdoc who made this called this molecule Barak. It was 2008. Barack Obama had just been elected. He was from Chicago, as it happens, and there was a torturous logic for this acronym based on the actual structure of the molecule. It doesn't matter. And then you could even kick it up another notch by shrinking the ring from 8 atoms to 7 atoms. If all the atoms were carbon, this wouldn't be stable at room temp, but sulfur is a little bit larger, so it relaxes the strain just enough so you can actually isolate and work with this molecule. And so what did we do with imaging? You know, we did a bunch of different experiments in different model organisms, but the one that lends itself to a visual is the work we did in developing zebrafish. Zebrafish are translucent. They're really wonderful for optical imaging. They will develop from the single stage embryo to a young adult fish in a matter of 5 days, very quick. And you can witness the entire developmental program under a fluorescence microscope with your naked eyes. It's quite spectacular. And what we discovered is that you can basically fertilize and then grow these embryos in media containing azidosugars, and the cells of the developing embryo will take up those sugars, integrate them into cell surface glycans, and now we could do these, what we called copper-free click chemistry, with cyclooctane fluorophore conjugates and put different colored dyes on the sugars at different time points during development. So, you know, you could do this once and then rinse off the embryos and then label them again with another azidosugar, and different azidosugars could be labeled with different fluorophores, and we'd end up with these rainbow fish where the different colors reflected glycans that had appeared on different tissues at different time points. And just to show you what that looks like under a microscope, here's the head of a developing sort of 5-day-old zebrafish. And this particular fish had been labeled at 3 different time points with 3 different sugars and 3 different colored fluorophores. And you can see how, you know, the different colors are distributed differently on various cells. For example, cells here in the sort of nasal cavity and the sort of upper respiratory tract, if you can call it that, you know, are very rich with sialic acids. They're mucosal tissues, and you can see that with the red coloring. Whereas the skin cells, the epidermis, you know, they were rich in a different kind of sugar. You can kind of see that too. And we could even watch the rearrangement of cell surface glycans during cellular processes like mitosis. Here's a 24-hour-old embryo where there's a lot of cell division going on and a lot of cell fate decision-making going on. And we noticed that a particular sugar that we introduced into cell surface glycans was being systematically redistributed late in the cell cycle every time a mother cell divided to form 2 daughter cells. Here's a blow up of a frame from one round of mitosis that we happened to capture. I don't know if the room is dark enough for you to really see this, but each frame is the exact same frame, but with different colored fluorophores through different filters. And so over here, we have a fluorophore on the DNA. So you can see the chromosomes are duplicating and segregating during this round of mitosis. This is the exact same cell undergoing cell division, but now the fluorophore is on a membrane protein. So you can kind of see how the membrane, you know, the mother cell is dividing, a new membrane fills in the gap between, and now you have 2 daughter cells. And here, for the first time, we could actually look at the sugars as they're changing during this process. And this is where we notice this kind of buildup of intensity of these particular sugars right at the junction between the 2 cells as they divide. And it turns out the sugar that we had labeled was contributing to the kind of physical separation of these 2 cells. So that was sort of fun to see. And anyways, we published a bunch of different imaging studies using this copper-free click chemistry. And then I'd say the field kind of took on a life of its own. As soon as we started publishing on what we were doing with glycans, other labs realized that you could use the same platform to image your favorite biomolecule. It could be a protein, it could be a metabolite, it could be a lipid, right? Whatever you happen to study. So people started putting azides into amino acids and feeding them to cells and imaging proteins as they were being translated off the ribosome. People started putting azides into nucleosides and labeling DNA and RNA with azides and then clicking on their probe of interest. People did it with lipids. You could actually watch lipids moving around in the cell because you could click probes onto them. And even drug molecules. You could put clickable functional groups onto drug molecules and then track down all of their targets and their off targets using these in vivo chemistries. So it turned out to be quite useful just as a platform for biological research. Nowadays, I'm most excited about the sort of translational use of bioorthogonal chemistries. They have been picked up by the biopharma industry and used in some really creative and interesting ways. It was mentioned that I have a personal interest in antibody drug conjugates, which I'm going to mention now in the last few minutes of the talk. And I have two companies that are using bioorthogonal chemistries to make these really exciting medicines. One of those companies is Carolyn, and the other is Firefly Bio. But all these other groups, companies, institutions, on their websites are advertising their use of bioorthogonal chemistry to make really complex medicines, often bioconjugates, that would be hard to make otherwise. And then there are companies that have right now vaccine candidates in clinical trials, like Vaxcyte has used copper-free click chemistry to make conjugate vaccines. Ace Podia is using cell surface copper-free click chemistry to make a new brand of off-the-shelf CAR T therapies. They're in the clinic with one of their products. And then this company, Shasky Pharmaceuticals, is actually performing bioorthogonal chemistry in the body of human patients to target drugs to tumor tissues. And so they're in the clinic as well. But just to sort of share the antibody drug conjugate craze with all of you, any of you in the practice of oncology have heard of ADCs. They're probably the fastest, kind of growing pipeline for oncology products right now. They are molecules that consist of an antibody that binds a tumor-specific antigen, and to which is chemically conjugated a toxin, if it's an oncology application. And the idea is that the antibody delivers that toxin only to the cancer cells, or preferentially to the cancer cells, and keeps the toxin away from healthy cells. So these are prodrugs of sorts. They need to get internalized into, whoops, what did I just do? There was some slide change, sorry about that. They need to get internalized through the endosome-lysosome pathway, where the antibody gets degraded and the toxin gets released. And then the toxin is inside the cell, where it can damage the cell through a variety of different mechanisms that lead to programmed cell death. If you look at the anatomy of an antibody drug conjugate, it's as you would imagine. There's the antibody component, there's the drug component, there's a linker that connects the two. And since the 1980s, there have been a lot of technical advances that have brought the field to where it is today. And most of those technical advances have focused on, how to make better antibodies. We're pretty good at that now. Okay, we make them high affinity, we make them human or humanized. There's been a lot of work in figuring out what are the best drugs for these ADCs. They have to be super potent. They have to have certain physical qualities. And most recently, I would say the innovation has been around the linker. It seems like it's an inglorious piece of spaghetti connecting these two exciting components, but actually, I would argue that having the right linker with the right properties makes or breaks the ADC more so than the antibody or the toxin even. And where we thought bioorthogonal chemistry could be especially useful was in the exact way that this chemical part, the linker and the drug, gets attached to the biologic part, which is the antibody. So the more sort of classic ways of making those connections between the chemical part and the biologic part are embodied in what I think are the two, kind of inaugural FDA approvals that really launched the field as we know it today, which was the approval of Seagen's drug Adcetris and Genentech Roche's drug Kadcyla. Okay, Adcetris is used to treat certain leukemias. Kadcyla is used to treat HER2-positive tumors, like breast tumors or gastric tumors. And the way Adcetris is put together is through the attachment of this is the toxin part, this is a linker part, but the chemistry involves cysteine side chains on the antibody, which can be exposed at the hinge region through doing some reductions on the antibody. Kadcyla, by contrast, is made by linking the drug linker part, the chemical part, to lysine side chains on the antibody through an active ester intermediate. And the problem with these two chemistries is they lead to heterogeneous products because there's a lot more cysteines and lysines on the surface of the antibody than you actually want to chemically react with your drug. And so to manage this problem, what these companies do is they'll take the antibody, let's say Kadcyla production, you've got all these lysines on the antibody. You really only want single-digit copy numbers of your drug on the antibody because otherwise you'll ruin the antibody. But you've got 75 lysines, so you want to react only 3 or 4 of them with your drug. So what you do is you kind of choose a stoichiometry of the drug to the antibody so that on average, you end up with 3 or 4 drugs per protein, but it's a statistical mixture, okay? And if you took a mass spec, as Immunogen and Genentech published, of the product, you'll see that there's just like this mixture of different molecules with different drug antibody ratios, and you get this heterogeneity. So on average, Kadcyla has 3 drugs per antibody, but the reality is there's something like 50,000 different molecules in a vial of Kadcyla, and all you get is the statistical average. So you can't optimize this drug. You get what you get. It's a big mixture. Some of the molecules in there are the good ones, others are the bad ones. You're stuck with all of them, okay? We thought you could do better with bioorthogonal chemistry, because if you could introduce a bioorthogonal group site-specifically where you want it and in the copy numbers you want it onto your antibody, then you could direct the chemical attachment of your probe explicitly to these functional groups and not worry about how many lysines, how many cysteines, because this has nothing to do with the natural side chains. This is a new chemistry that you introduced. And we figured out how to do that using a technology that now we call the aldehyde tag method. So there's an enzyme called formyl glycine generating enzyme, FGE, which naturally does a post-translational modification of cysteine, converting that side chain to an aldehyde group called formyl glycine. This enzyme is in our bodies, it's in all of our cells, it's in the endoplasmic reticulum, it has a natural function, but it'll recognize a short 5 amino acid motif that you can clone into any protein of interest, we discovered. And so what we do is we clone that sequence where we want that aldehyde to be in either the heavy chain or the light chain gene, and then we express the heavy chain and light chain in CHO cells or HEK cells together with a little extra FGE, and those cells start cranking out the antibody where there are now aldehydes at the site that we had encoded with this motif. The aldehyde is bioorthogonal on the surface of a protein. There's no aldehydes in any of the natural side chains. And we invented a chemistry that we call the HIPS ligation that allows one to specifically attach anything of interest to these aldehydes, like the linker in a drug for an ADC. And if there's any hardcore chemists in the audience and you're wondering, how does that HIPS reaction work? Here's the mechanism, okay. So we commercialized this at a company called Redwood Bioscience. That company was then acquired by Carolyn, which was acquired last week by Novo. Okay, so now we're going to be part of the Novo empire. But basically what we do at this company is some partner will give us the sequence of the heavy and light chains of their favorite anti-tumor antibody. We then, clone in our motif, express that antibody with as many aldehydes as you want at the sites you want them, click, on the drug linker through that HIPS ligation chemistry and make ADCs that are tailored to the needs of the particular indication. So that's an example of how we use bioorthogonal chemistry to make better ADCs, where we control the chemistry and the structures. But there's a number of other ways that you can use this chemistry to make different therapeutic modalities that would be really hard to construct just using kind of regular off-the-shelf chemistry. So, if you're interested, you can kind of look through the companies that I have formed online that use bioorthogonal chemistry in different ways to fuse interesting molecules together to make new therapeutic modalities. But I think there's really a huge future for this just in terms of making new drugs, regardless of whether they're made outside the body or in the body. So with that, to leave time for questions, I'll just thank all of my past and present coworkers. I gave you this sort of epic story of bioorthogonal chemistry from back in 1996 when I started my job at Berkeley. Too many people to thank, because there's so many people that contributed. But after the Nobel Prize announcement, my alumni somehow found each other all on LinkedIn and through the email network and so on, and arranged a Zoom celebration. And we managed to get about 150 current and former trainees on Zoom at that time from all over the world in weird time zones and so on, to drink a glass of champagne. So here we are, many of us at least, celebrating that moment. And with that, thank you very much, and I'm happy to entertain questions. Yes. There's a microphone coming your way.
So much for a wonderful talk and for the wonderful work that you've done over the decades now. It's been known, for I think since the 80s that people with chronic disease, particularly chronic kidney disease, express increased sialic acid residues, like on like oropharyngeal epithelial cells. And I just wondered if it's either known or whether you could speculate about why that occurs. Like, what are the mechanisms that cause that to happen?
Well, so I wasn't aware of that observation, but I can tell you what we have learned in the context of tumors, where this hypersialylation phenotype is well documented. It has to do with immune modulation. So there is a family of immune modulatory receptors on immune cells, and we call them the SIGLEC family, which stands for sialic acid binding Ig-like lectins, SIGLEC. This is now actually a big focus of my lab. These receptors are found, there's 14 members of the family in humans. Different members are found on different immune cell subtypes. And about 2/3 of the SIGLEC receptors have signaling domains that are similar to PD-1, the T cell checkpoint receptor. So they're inhibitory receptors. And when they engage ligands on targets, that delivers an inhibitory or suppressive signal to the immune cell. So whenever I see a setting where sialoglycans are upregulated, I can't help but wonder if that is in response to chronic immune activation, and it now is part of a resolution program. So that's what's happening in tumors. Tumors that are hypersialylated are able to suppress any infiltrating immune cells, and that is a survival advantage. So it's a phenotype that arises through natural selection. And conversely, if you block SIGLECs or the sialoglycans they're binding to, you can potentiate immune reactivity against cancer. And in fact, 2 of these companies are making immune therapies that target that axis of biology. So that would be Paleon. Paleon has 2 assets, one of which is now in clinical, a phase 1/2 clinical trial, and Stanford is one of the sites actually for the phase 1 part of that. And then this company, Valora, is also making drugs that target that axis in a different way. But so since we know sialoglycans modulate immune cell reactivity, I can't help but wonder in this setting whether that might be part of the story. So is there an inflammatory component to that disease?
Mucosa stickier, and so it's been known that they gram-negative rods normally don't colonize the oropharynx of healthy people, but yet a lot of chronic disease, particularly CKD, we do see it. So yeah, it seems like maladaptive, but
okay. Well, I have to read about that then. Maybe there's something we can do to help understand that better.
Hi, that was great. Really enjoyed it. 2 questions. One is there a way to unclick? So you deliver and then you unclick because you don't always want it there. The second question is, like, it's amazing to see the history and the story of how specific and deliverable and fast you've made it. Can you think forward 5-10 years? Like, how much better can it get technologically?
Yeah, there have been some really great advances, not from my lab, from other labs. I don't have it in this deck, unfortunately, but there's a chemistry that people use now, and it's the one that's being used in humans by Shasky, called the tetrazine ligation, which is yet another, like, maybe order of magnitude faster than the fastest azide alkyne cycloaddition that we developed. The tetrazine ligation involves other functional groups. One is a tetrazine, so 6-membered ring with a bunch of nitrogens in it. And then the other is a trans-cyclooctene. It's actually a strained alkene. And so that chemistry was developed simultaneously by 2 labs, one in University of Delaware and the other at UC San Diego. And they kind of took a page out of our playbook and then made it even better. So not only is that chemistry even faster, also there's a way to do what's called click to release. So people have figured out how to use that chemistry to unclick things. And I think there could be some applications of that. So, and I can't take credit for that, but I'm really excited about that chemistry. And we use it in my lab too for various reasons.
Can you deliver these chemicals inside the cell using like liposomes and then have the disease-specific reaction occur intracellularly?
Yeah, you can. Yeah, we have made renditions of these reagents that are membrane permeant, and they can get into cells, and you can have chemistries take place in there. And one of the companies I'm working on now that's very in the early crystallization phase is a company in which we hope to assemble drugs in situ using inside the cell click chemistry, right? Where you take the two components independently that on their own can get into cells, but the product can't, right? So for drugs that really they won't be orally absorbable, they can't get into cells. If you could just make them inside the cell with the components, then maybe you'll do better that way. So that's sort of what we're doing. Yeah, we'll see how that goes.
Thank you for the wonderful talk. I was curious to know, is it possible to draw correlations between glycans and their distribution and dynamics and DNA sequencing, maybe by using your imaging methods and altering DNA and observing the changes?
It's a really interesting question. You know, one of the reasons glycobiology is relatively underpopulated in the world of biology is that glycans are not primary gene products. And so their relationship to the genome is much more distant, right? So if you know the genome sequence, you can predict the proteome sequence and the transcriptome sequence, but you can't easily predict the glycome from the genome. And when I say easily, it's not like there's a one-to-one relationship. Glycans are the product of biosynthesis that involves hundreds of genes. So hundreds of genes encoding hundreds of enzymes, all working together to make this collection of glycans. And it's so complicated, that network is so complicated that people have not really been able to make these predictions simply by sequencing the genome. Having said that, in the era of machine learning and all that, maybe there's a way that you could train an algorithm with as much imaging and glycomics data as you can collect and correlate that with genomic signatures and make these predictions learnable by a machine, if not by a human. So that is something I think people are interested in. Oh, there's an
there is an online question from Dr. Golden asking a question about something near and dear to my heart as a geo-oncologist. For very aggressive prostate cancers, are there even more sialic acids attached to the cancer cells? And I know some interesting data there.
Generally, I can't, I'd have to go back and look through the correlative clinical literature, but pancreatic cancers in general are known to be hypersialylated. They fall into a category where they often are, you know, very rich in sialic acids. Whether people have bothered to try to correlate the extent of hypersialylation with the aggressiveness of the cancer, I don't know. But, you know, I think we all know that the first generation immune therapies that target the T cell checkpoint receptor or ligand, so the anti-PD-1 or CTLA-4, those types of therapeutics, they generally fail in pancreatic cancer. They fail in breast cancer. They fail in ovarian cancer. They're not effective at all in those settings. And one hypothesis we have is that those are tumor types that tend to be very highly sialylated. So maybe the Siglec axis of immune modulation is a more important axis in those disease settings. And so those patients wouldn't respond to PD-1 therapies. Maybe they would respond to an anti-Siglec or an anti-sialic acid therapy.
Right, so opening the door to therapeutics in cancer.
We hope, yeah. That's another talk. Invite me back and
That sounds
I'll talk about
that. Okay. And with that, it's 9 o'clock. It's
All
hard
right.
to believe our hour.
Thank you very much.
Thank you so much.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.